So I will just jump in now. So the first comment that is important, and this actually was a discussion that I actually had with Paul Root back when he brought this up, was that he had some rules of thumb for older products that he had used most of his entire life. But we concluded, even without any data, we concluded that the likelihood of rules of thumb for organophosphates is not likely to be appropriate and useful for a lot of the new materials we have, whether they are the spinosins or ivermectins or neonicotinoids. And there's diamides are an increasingly important class of materials. So we really needed to choose compounds from each of the important classes of materials and test those and make some assumptions that what we learn from the selected material would be representative of other products in those classes. So you'll see that that's what we did. In fact, the additional thought is that there are more biopesticides available today than there were a decade or more. And so we also want to learn about the rainfastness and performance attributes of biopesticides as well. But maybe the starting comment, so that sets the stage for how to think about these data, is that there's a lot that happens after you make a pesticide application. Certainly, the way in which you spray is going to impact deposition, right? Coverage and deposition. So there's variability that can occur in relation to spray coverage and deposition. But then even after a product has been applied, we know that immediately there's photodegradation that's beginning to eat away at the residual toxicity of the materials that we're using. But then also we've learned over the last decade or more that many products have penetrative attributes, that they can move into plant tissues. And the way that products move into leaf tissue is different than the way that they move into fruit. And once those products are into tissue, what happens at that point can also vary. There's a lot of materials that can have translaminar movement from the surface to the underside of a leaf. But also there are materials that have acropedial, basopedial movement. Acropedial means the product moves in and it is picked up by the xylem and moves out to the endpoints of the leaf. Those are often like neonicotinoids that are more water soluble. And there's also products that move in the phloem back into the sinks of the tree. A product I can think of in particular would be like Movento, that it actually, if you apply it to the foliage, it moves back down into the roots to kill all the apple leaf. So what's my point? That's a whole presentation in itself that we're not going to do today. But my point is that all those things influence the result of rainfall on the product and the performance that you're expecting. This is a summary of some of those attributes that I just talked about. If there ends up being handouts, this could be a one-pager if people wanted to bring it home, but it basically summarizes what we know about the systemic characteristics of all these classes, organophosphates and pyrethroids and neonics in foliage and in fruit. And in general, what we see is that there's lots of materials that are primarily surface materials. They may move into the cuticle, but they're not going to be systemic in the way that we think of other products. And then you've got products like neonicotinoids that they are not only translaminar but also acropedial and systemic as they move. And then like ivermectins, spinosins, and diamides, they're translaminar, that they're going to move through that leaf tissue, but not be quite as mobile as neonics and products like Movento. All right, so that the reason I put that slide up there is that it's important. It really has a big influence on rainfastness of products. I'm actually going to skip this slide. I think I basically have covered it. Maybe the only comment I'll make is that drying time and additives also can have an influence, but that's beyond the scope of today's presentation. But we certainly can talk about what we've learned in that respect. All right, so this study for this master student really had two primary questions. One was, what are the effects of various rainfall amounts to insecticide control against SWD in cherries, both the effect on adult toxicity as well as infestation by larvae. And then the second question was, well, when we test these different rainfall amounts, we also want to know how they affect the surface and subsurface residues on leaves and on fruit. And so we looked at both the effect that rainfall has on the pest control, but also on the residues. And I'll tell you that if your thought is that, well, wouldn't they be the same, that as you lose residue in a linear way, you're going to lose pest control, I would say, no, it's not the case. It's not nearly as straightforward as that. It's a more dynamic process, and we will see that when we get into the data. So here are the products we chose. We just tried to choose a representative material from the major classes of insecticides that are important for spotted wing Drosophila control in cherries. So we had Phosmet as our organophosphate, and applied at 2.125 pounds per acre. This is Grandivo. I wanted to have one biopesticide included in this study. And over the years, I've had some good performance experience with Grandivo and spotted wing Drosophila. And I've also had some years where it didn't perform as well, so we wanted to include it. In the diamide class, Exeril was the product that we chose. In the neonicotinoid class, we chose Assail. For the pyrethroids, we chose Mustang Max. And then for the spinosas, Delegate. So those were, we thought, were the best choices that would be representative of those classes. And then the product rates were generally what's labeled or recommended. The sequence, we have a sequence of steps that we used for, oh yeah, I wanted to add that in the orchards at Trevor Nichols Research Center, we bagged the fruit before the study began, because again, we wanted the fruit to be at a stage where once the study began, we would have robust infestation in the fruit, but we didn't want any infestation before the study began. So we bagged the fruit, and then at the time that the study began, we pulled the bags off and we sprayed with an air blast sprayer, and then the spraying occurred, and then within 4 hours we picked the fruit and began the bioassays. So in the bioassays, we first simulated rainfall, and we had a 0 rainfall treatment. We had a 1/2 inch and then a 1 inch treatment, and we picked fruit clusters from the trees that had been sprayed. We put them in these cute little bioassay chambers, and then we put 12 adult spotted wing Drosophila, 6 females, 6 males, and then we measured. Ignatius did a little bit more intensive measurements over time, but essentially after 5 days of exposure, then the flies were removed, and then we were able to measure mortality of adults at that point. And then after an additional 4 days, we used the brown sugar method and looked for larvae in pupae. Parallel to that process, an equal or a parallel set of samples were taken, went through the same rain booth settings, but in this case, instead of going to a bioassay, we took those fruit and leaves and we sonicated them in acetonitrile, take off the surface residue, and then the rest of it was mashed and held in dichloromethane, another more aggressive solvent, and then worked with our pesticide analytical lab on campus to end up with parts per million of the various materials. Okay, so every slide is going to be the same, compound by compound. So I'm going to walk you through this. And I used to, when I showed some of these data at IPM school, I separated the residue data and in separate slides did the adult mortality. And I got input from people saying it was really hard to remember what the previous slide was. So I squeezed them here and put them both on one slide so that you can look back and forth. But I want to first just talk you through the residue data. So we have on the green side, the leaf residue data, the dark being the surface and the light color being the subsurface. And then on the red side, the fruit data, and the dark red being the surface and the subsurface residue being the lighter color. Okay? And then on the bioassay we'll get to next. Yeah, let's just hold that. We'll just start here. So if we first just think, I want to know what happened to the material first, then we can look at impact on performance. And this is phosmet, this is an organophosphate, and the first thing that we see fairly clearly is that imidan is highly sensitive to wash off. You can see in particular on the fruit that a half-inch of rain removed a very large portion, probably almost 90% of the residue, and then maybe just a little bit more when we went up to 1 inch. Imidan is a little bit more resistant to wash off for those residues that are on foliage. You can see this. There is a reduction. It's a significant reduction, especially when we get out to 1 inch, but not as dramatic as what's on the fruit. And if you ask, well, why is that? Well, the surface of that leaf is really quite different than the surface of the fruit, and there's a binding affinity that each compound has. And for the case of imidan, it's more resistant to wash off foliage than it is a fruit. So if you look only at that, you're thinking, oh my gosh, this rainfall is devastating to phosmet. But when we look at the adulticidal data, how many adults were killed, yes, there was a dramatic reduction in adult survival when we went to a half-inch of rain and 1 inch of rain. But remember, we're starting up here at 90-ish% mortality, and even after the rainfall, we're still somewhere in that 20-ish, 25% mortality. But no doubt, significant reduction after those rainfall levels. Okay, our pyrethroid product, Mustang Max. When we look first at the residue data, the most striking thing is that there isn't that same loss of parent material in the fruit or in the leaves. While I would suggest to you that while the product really is not a systemic material, when we're picking up zeta-cypermethrin leaf subsurface and fruit subsurface, really what that is it's bound well enough into that cuticle that it was not removed with the surface solvent that we took. I don't think you would want to interpret that as this is a systemic product, because pyrethroids are not generally systemic in that way. But it tells you that's probably, there's just a difference from the organophosphates in how much binding and resistance to wash off that you would expect. However, here's the part that's a little bit interesting, is that when we looked at the adult mortality data, there still was a significant reduction. And I was perplexed for quite some time. How do I explain this? What is it that we can take these data and help inform, help inform what's happening here. Now, the reduction in adulticidal activity wasn't as dramatic as we saw with imidan, but there's definitely significant reduction in adulticide activity. And I think really what it comes down to is that the exposure to rainfall is changing what in the academic literature is called bioavailability. That a product can be present, and it is. We detect the parent product, it's there. But the bioavailability has changed. I don't know details of what exactly that means, but I think that there's enough other publications out there that tells me that, okay, it's a really good thing that we did the bioassays, because if you only looked at the residue, you say, it's rain fast, we're good to go. But it tells us that, okay, there is a half-inch of rain, 1 inch of rain is going to significantly reduce the adulticidal activity of this product. Okay? Yeah?
When were those, how soon after application were those rainfall events?
Yeah, so the rainfall events were within 24 hours. Yep. So that's fresh residue. Okay, neonicotinoids. The sales, the product that we used, and we look at the residue, and what we see is that it is a fairly rain-fast product. Okay, so if you look just at the residue, and it has to do with the way that the product moves. I've done other work with neonicotinoids, and in other cases we've seen more subsurface than what this shows. But without getting into it, I think there were some methodology differences in how the student did this. But regardless, this is consistent with what I've seen in apples and blueberries and other cases. That the neonics, because of their penetrative attributes, they tend to be fairly rain fast, and we don't see the losses what you saw like with the imidan. However, when you look at the adult mortality, now what we see is that no, there's not a reduction in the adult mortality. But the problem is that a sale really is not a strong adulticide in the first place. If you use the sale for SWD control, if you've got performance, it's not because of the adulticidal activity. There's lots of ways that a product can protect your fruit, but this makes it very clear that the neonics are not doing it from their toxicity to adults, because it's just not the way they work. So those are the data. So my take home here is that, yeah, the sale is maybe of the highest rain fastness of the materials that we've seen so far. It's just not a very good adulticide. But there's more, so just hold the thought. Spinosins, we use Delegate, and what we saw was that Delegate at a ½ inch and 1 inch, it was the leaf tissue that had the greatest losses. And I've seen this in the work I did in apples as well, as that Delegate comparatively is more rain fast on fruit than it is on leaves. It just has to do with chemistry and what things tend to bind to. And so, yes, there's a reduction in residue on the fruit, but just like in other trials that I've done in apples, it's really what's lost on the leaves that is the more dramatic loss. When we look at the adulticide activity, Delegate is more active on adults than the neonics are, but nothing like what we saw for imidan, right up there at that 90 percentile. And the more statistically significant changes in adulticide happened at the 1 inch, not as dramatic as we saw with a couple other compounds. But there is an effect according to rainfall. The diamide, Exerl, residue data on fruit and on leaves shows it to be fairly rain fast. Numerically, we see this decline, but it did not actually sort out statistically on the leaves. But I think it's probably fair to say there was some loss on leaf tissue, but on the fruit tissue, it was even up to 1 inch of rain. It was quite rain fast. And then when we look at the adulticidal activity, there's actually a statistical drop from about 60% down to 20% at that 1 day, but we didn't see that same effect at 3 days. And I think it's just a reflection of some noise in the system in doing the bioassays. But my summary would be that it's just kind of a moderate reduction in adulticidal, but by no means a highly toxic adulticide product, not like what we saw for Imidan, right? We didn't have residue data for Grandivo because it's not a synthetic material that we could run through the equipment we had, but I did want to show the adult data where it was, even with zero rainfall, it's something similar to a sale that is not a very good adulticide, Grandivo, and there was some reduction in performance with a 1/2 inch and 1 inch of rain, but it wasn't statistically significant. This slide shows all the data for the control of the juvenile stages, mostly larvae and some incidental eggs and pupae that were picked up in the assay. This particular slide, though, I decided to focus on large larvae because it was the most predominant stage that was caught. What we saw first, looking at the untreated, is that we were getting somewhere like 35 to 40 large larvae out of each sample that was brought up to 9 days of exposure in that brown sugar method. And I'll point out again that the white bar is zero rainfall, the light blue is a 1/2 inch, and then the dark blue is 1 inch. And we look at the phosmet, and it matches up pretty close to what we saw with the residue in the adult data, that with no rainfall, it's excellent. And then there is a reduction in activity with the rainfall levels, but still, because it starts out so good, that even with 90% loss of residues at 1 inch, you're still having significant improvement over the untreated. So part of what we're learning is that, phosmet is highly sensitive to wash off, but even in the wash off condition, you still have some potency. Zeta cypermethrin, fairly similar, but just a little bit higher, and not statistically higher, but the pattern is just a little bit higher, but similar pattern to phosmet. A sale, again, pretty flat, really. Not the rainfall level did not make a difference in its protection against larval infestation. And that's, I think, because that product is systemic and has the curative action that we've seen in other crops and other pests. The neonics are going to move in and do a better job killing larvae than they do killing adults. And then here's Delegate, and there was an increase in larval infestation with rainfall. But again, it also has some penetrative attributes, not quite as dramatic as a neonic, but there's some movement of Delegate into the fruit tissue, and we get some larvicidal action. Here's the diamide external, and this, I think, suggests that there is some larvicidal action from external, because after the rainfall, we did not see a statistical difference in the number of larvae came out. Very, very flat, kind of similar to a sale in that regard. And then the last one is Grandivo, where there's definitely an impact of rainfall, especially when you get 1 inch, you're going to lose performance with a product like that. Well, those are the data that I have for today, and the conclusions that Certainly, for Imidan, it's highly sensitive to wash off, but it has highly potent adulticidal action, and so the remaining residues are still going to have some effect on SWD. Delegate or spinetoram, residues were sensitive to wash off, but in particular, the adulticidal action is reduced. Mustang Max, adulticide action in infestation was sensitive to wash off, but the residues were more resistant. We already talked about Grandivo, and we talked about the neonics, that they're actually probably technically the most rainfast of the products we tested. It's just that they're probably under high pressure. It's probably not a standalone product, but it might be one that could be used strategically with an adulticide. And then the diamides, the action was affected by rainfall, but the immature stages were fairly resistant to the effects of the washout. I updated the chart in the fruit management guide to reflect the data that we have, and as I mentioned, we did more work this year with testing 1/10 of an inch, 1/4 of an inch, and 3/4 of an inch. So we have more rainfall settings now. Those data will go into this modeling effort that Keith Mason is leading, and it will go to inform future information in the fruit management guide. And then here's the crew at the Trevor Nichols Research Center. Here's Ignatius, the master student. He's graduated and gone home to Indonesia. Never wants to have another Michigan winter, but we appreciate his contribution. And then I want to acknowledge the ROAR grant that funded his work. There's been additional work, especially block grants that have contributed, and then all the companies that made their products available for testing. That helps us learn and be better at what we do.